Hello everyone, welcome to the world of clinical histology. In the last section, we introduced the cavern epithelium and have also touched the nephron tubules in the kidney and cavern epithelium in the intestine. We mentioned that they both have a mechanism to increase surface area. In the pseudostratified ciliated columnar epithelium of the trachea, you can find motile cilia. These are all the unique structures of the epithelium tissue. So that's what we're going to talk about in this section, the specialized structures of epithelial cells. In this section, we need to be able to list the names of these specialized structures, describe what the distributions and functions are, and how they achieve these functions. In the first section, we learned that due to polarity, epithelial cells are divided into three planes: the free surface, the basal layer, and the lateral planes. Let's talk about this one by one. Let's start with the free surface. We have a picture of the simple columnar epithelium of the small intestine, and on the right is the proximal tubule of a kidney nephron. The arrows are directed to the free surface. We can see the morphology of these two simple epithelia under the light microscope. They have the same specialized structures called microvilli. These protrusions effectively extend the outer surface area of the cells. For example, the microvilli of the small intestinal epithelial cells increase the free surface area by 30% to 40%, which is beneficial for the absorption of nutrients. The electronic micrograph shows the microvilli in detail. In fact, there are extensions of the cell membrane that are supported by interior microtubules. Do you still remember this picture? It's the pseudostratified ciliated columnar epithelium of the trachea. Let's focus on the ciliated cells. Under the light microscope, the cilia are thicker and longer than the microvilli. In the middle scanning electron micrograph, we can see that these cilia have a structure like reed ears. Each cell has 200 to 300 cilia. Unlike microvilli, cilia consist of tubulin, connexin, and dynein. Dynein is a micromolecular ATPase, which has a similar function to myosin. The cells of patients with carcinogenous syndrome cannot synthesize dynein, leading to the respiratory epithelial ciliary motility dysfunction. The patient cannot clear inhaled dust and pollutants and are thus prone to developing bronchiectasis and sinusitis. In addition, the sperm tail also have a specialized type of cilia enable it to move. So men with carcinogenous syndrome have reduced fertility. Our understanding of the human body cannot stop at the level of cells and tissues, but also at the molecular level. We find that the loss of certain protein during translation of the same gene may cause the dysfunction of different tissues and organs. Therefore, if the mucous layer is lost, the movement of the cilia also stop. The human body is well designed and requires the effective coordination of various systems and organs to maintain its normal functions. We should keep the concept in mind all along. Have you ever thought about how skin epithelial cells acquire properties such as anti-friction, resistant to certain pH, and fending off pathogenic microorganisms? It's possible to achieve this by stacking the epithelial cells together. Just like the rice in the picture, They are also heaped together, but they will spread out once we let go of it. How about building a house? How do I keep the bricks together? With cement, right? So we can infer that epithelial cells may have a similar binding pattern. To prove this assumption, we need to use electron microscopy to study these specialized structures between cells. These are the intercellular connections present on the lateral planes and involve tight connections, intermediate connections, desmosomes, and gap connections. This is just a brief introduction with diagrams to give you the overall concept of connections between epithelial cells. The specific protein compositions or how these proteins connect with each other are more for research, since their clinical applications is relatively limited. You can read about this after class if you are interested. First, we have the tight junctions. Let's look at the following two schematic diagrams. The tight junction often appears on the lateral side of epithelial cells. A specialized protein forms a closed lock between the two adjacent cell membranes. Their joining membranes are closely stitched together as delicately as a saw dress. This specialized feature is one of the most important structural foundations for epithelial tissue to protect and segregate themselves, and also forms the frontline barrier for our body and important for maintaining polarity. This electron micrograph shows a cross-section of the epithelial cell tight junction. In addition to the tight junction, desmosomes also play a role. These are especially advanced in the stratified squamous epithelium of the skin and esophagus, which are vulnerable to friction. Let's take a closer look. The picture on the left shows the desmosomes seen under the transmission electron microscope. In the lateral plane of the membrane, there is a high electron density attachment plates, and keratin filaments are attached in this region. In the small gap between the two cells, one end of the transmembrane protein is connected here, and the other end is connected to the attachment plate. In the skin and esophagus, epithelial cells are connected securely with each other by desmosome junctions. For example, the diagram here shows that in the myocardium, the presence of tight junctions allows the myocardium to maintain efficient contraction and relaxation throughout life. In the picture, we can also see other connections between cardiomyocytes, including intermediate and gap connections. They are closely related to signal transduction and substance exchange between cells. So we can see that intercellular connections are not unique to epithelial cells. There are also different types of connections within muscle or nerve tissues. Cell adhesion is mediated by catenins, transmembrane glycoproteins that bind adjacent cells. The transmembrane gap junction proteins, connexins, form hexameric complex called connexons, each of which have a central hydroxyphilic pore about 1.5 nanometer in diameter. Gap junctions are critical for electrical transmission because the connexin channels within and desmosomal connections allow ions to pass from one cell to another. Electrical transmission is much faster than chemical transmission. This is how all the heart muscle cells coordinate to contract together. In the first section, one of the characteristics of epithelial cells we mentioned is that there are no blood vessels. The nutrients for the epithelial cells come from the connective tissue below. These nutrients need to traverse the basement membrane to reach the epithelial cells. We know that the covering epithelium acts as a barrier and has a low permeability, which means the basement membrane must be selectively permeable. How does the basement membrane achieve this function? This is an electron microscopy section showing the detailed structures of the basement membrane. The part nearest the epithelium is called the basal lamina, produced by the secretions of epithelial cells. The reticular lamina is formed by and connected to the connective tissue. These structures are made up of a variety of proteins. We are familiar with some of those, such as collagen and laminin. These proteins interweave into nets to form a semi-permeable membrane-like structure that facilitates exchange of selected material between epithelial cell and connective tissue. At the same time, they also have a binding effect to connect the epithelial cell with the underlying connective tissue through hemidesmosomes. This picture shows another function of the basement membrane. Certain proteins regulate proliferation, differentiation, and movement of epithelial cells. In the previous section, we mentioned that stem cells in the basal layer of the skin can differentiate and move upwards. If something goes wrong with the signaling molecules, abnormal differentiation and proliferation will lead to tumorigenesis. When a malignant tumor develops, the basement membrane also prevents the cancer cells from downward infiltration, but this function relies on the effective roles on lamina, cathepsin, and other proteins. The products of some cancer cells can break through the basal layer barrier by secreting proteases to disrupt the basement membrane, resulting in extension and metastasis of the malignant tumor. You are going to learn more about this in pathology. Today, we have talked about the specialized structures of epithelium and other types of cells. We also discussed the relationship between these structures and cellular functions. In the next section, we will continue to describe the characteristics and roles of the connective tissue beneath the epithelium. Thanks for joining.